So in this diagram here, you have a membrane and you have solute and solvent. The solute is are represented by these round molecules, and the solvent is by these little pluses. The solvent here is water. The key tells you what's going on. This is the left side, obviously. This is the right side. And then they ask you these questions. You should be able to answer those without too many problems, and no one has posed any issues with it. I would consider something that we discussed in class, just to make sure you're getting this correct here, is that when you're talking about here the number of water molecules, all you're going to do is count them, right? Number of water molecules, you're going to count them. Then when they ask you about ratio, I want to remind you that a ratio is pretty much a fraction, all right? So when you're looking at a ratio, you're saying I'll do the one on the left for you. It's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. 24 water molecules, and it's so there's ratio of water to sugar, so it's 24 to 6, right? 24 to 6. So when you're talking about that's the ratio, right? So that's water molecules, that's the solvent, and this is sugar. And when they say ratio of water to sugar, the 2, when you're talking about a ratio, ends up being this colon. Let me erase this colon right here. Put a dash instead. So when you're talking about a colon, that you can translate this. Sorry, I'm really still sick here, but this 2 gets translated into a colon. Now, when you change this into a fraction, okay, when you go ahead and change this into a fraction, so when you change this into a fraction, what you get is 24 water divided by 6 sugar. Now, if you go ahead and made you could put that in a decimal form. So that's 24 water for every 6 sugars. So that would be 4 water molecules for every 1 sugar molecule. And that's the advantage of using a fraction, because then you can simplify. 6 divided by 6 is 1. 24 divided by 6 is 4. So 4 water molecules for every sugar molecule. Now, we normally don't write concentrations this way. The best way to write concentrations is solute over solvent. So it would be really 1 solute over 4 solvent water. Still, you have 1 sugar for every 4 water. Right? And so you can make that into a decimal if you'd like and put 0.25. So that would be the concentration on one side of the membrane. And I believe that was, I can't even remember anymore. I think it was the left is the side we did. So that would be 0.25 in decimal form, or 1 out of 4. 1 to 4 would be the ratio 1 to 4. But since they're asking you the ratio of water to sugar, you have to put water first. So it'd be 24 colon 6, right? So it would be 24 to 6. And then on the right side, you go ahead and count. And I think you're going to come up with some number, and you have to decide which one's bigger. Which side of the membrane has a higher concentration of sugar versus water. And that's where putting it in this form, making sugar and then dividing it by water and coming up with a decimal really helps. Because you're putting it the solute over solvent, you compare one side to the other. And you'd expect that if the solute is smaller, if the solute is bigger than whichever side, left or right, where the solute relative to the water, so the division, the ratio, is bigger, sugar solute to water is bigger, then it's more concentrated, right? So when you're talking about concentration, you're talking about comparing one side to the other. You're talking about considering solute divided by solvent, right? So I'm just going to go ahead and Oh, I guess I should have filled it in all the way and said 24 water, 6. And then if you did the same here on the right side, I'm going to go ahead and do it then. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. You have 15 on this side, but then how much water do you have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 13. Right? So now it's 15 to 13. So then you have to ask yourself, where is sugar the highest? Well, sugar is the highest here. But it's not about where sugar is the highest number. The question is, where is sugar the highest number relative to water? So where is the ratio higher? And it might still be the right side. So we know what this is. On the left side, we know what it is. But on the right, we're going to go ahead and make a division, right? We're going to go ahead and divide. But now this, instead of 24 divided by 6, you're going to say 15 divided by 13. 15 water molecules for every 13 sugar molecules. Now, obviously, sugar's higher. So if we take a look at what we did, I did the same thing down here that I did up here, where I took water divided by sugar, I got the number 4. Well, water divided by sugar gave me the number 1. So which side is, but here in both cases, what I'm looking at is where this one's obviously water's higher than, because here I have 4 water for every sugar. 4 water for every sugar. Where down here I have 1.1538 water for every sugar. Right? So this is, when I'm looking at this here, and I'm comparing these two numbers, obviously water, I have more water for every sugar up here than I do down here. But as I said, with concentration, we like to look at sugar divided by water, and that's 0.25. That makes it easier to understand where's sugar the highest, because you're dividing it by the solvent. So it's how much sugar is dissolved in water. So really, that's the standard we use. So that would be sugar and water. That would be 0.867 versus 0.25. So obviously, sugar is more concentrated. Sugar is more concentrated down here. So it's the right side that is more concentrated than the left. More concentrated solute. So the solute is more concentrated out here. There's more sugar for every water. You can kind of look at and see that there's more sugar than there is water on this side. So you have 0.867 because now you have a number that verifies it, where this number here, 0.25, tells you that it's smaller than 0.867. So when you're doing this kind of problem, you got to consider, guys, that you're looking at sugar divided by water, and asking yourself, what's the concentration that I'm dealing with at this juncture, at this point? And where's the sugar the highest? Obviously, in this case, the right side is more sugar for every water molecule. Even now, that makes sense though. That if you look at and reverse it, that this number's smaller, because here you're looking at how much water you have for every sugar. You have less water for every sugar, so that means you have more sugar than you have water. It's kind of what we had discussed in class. They're reverse of one another. So when you're talking about ratios, it's important to know what you're looking at, which one's on top, and what that fraction is actually telling you. So this 4 is telling you have 4 water molecules for every 1 sugar molecule. Here you have 1.158 water for every sugar molecule. That tells you it's more concentrated. However, it makes more sense, I think, to me intuitively, and certainly as a standard, to look at sugar divided by water. So here it's 0.25 on the left, and here's 0.867 on the right. And so obviously, you can turn it into, let's leave it at that. Yeah, 0.867 is bigger than 0.25. So at that point, you have to consider 0.867 is the more concentrated. So no matter how you look at it, one way or the other, the right is more concentrated than the left. Now here you have this membrane, and again you have the solute and the solvent being the pluses and the solute being the circles. You can discuss then what's inside, which side of the membrane is more concentrated sugar solution. Again, you're looking at sugar divided by water. So sugar, the number of sugar divided by the number of water. Right, so you can go ahead and do that math, and you should be able to answer that by counting and deciding which side of the membrane has more concentrated sugar solution. So more sugar for every water. So obviously the higher the percentage. So, I'll do the outside again for you. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So that's 15 water. And 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 sugar. Add to 15 water. So that's a concentration of 1 sugar for every water molecule. So then the next question is, what are you going to do if you know that the outside's 1 sugar for every 1 water molecule? What do you think it is on the inside? You can make that count. I think you'll see just by looking at it, there's obviously more water than sugar inside. It's not equal. Outside it looks like it would be equal, and it is, but inside it obviously isn't. So there's obviously more water inside, I think. You can make the count, and you can come up with a difference. You can come up with a number yourself. So that would suggest that outside you have more water. Inside you have more water than sugar. You have more water than sugar. So then now you have to consider whether this stuff is a higher. So that means that inside or outside it's more concentrated. The solute is more concentrated, right? So there's where inside you have more water than sugar. Outside you have equal water and sugar, 15 to 15. Inside is going to be the water is going to be much larger, so you're going to have a smaller fraction. So I'm going to go ahead and count these and do it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 19, 20, 21, 22, 23, 24. So you have 24 inside, right? 24 water molecules. And sugar molecules, you have 6. So that gives you, so sugar to water. So 6 divided by 24 is not 1. 6 divided by 24 is, it would be 0.25 sugar, so 0.25 sugar for every water molecule. All right. So for every sugar molecule, yeah, if you wanted to look at percentages, you could, but you'd have to figure out the total, right? To get a percentage, you'd have to figure out the total, and it's probably easier just to go ahead and use these fractions. So inside it's 0.25. Outside it's 1. The ratio of sugar to water is 1. So where is sugar more concentrated? Where's the sugar? Where's it higher? Where's the number? Where's this fraction, this ratio higher? Well, it has to be outside, right? See, the sugar water is more concentrated outside than inside. So we'd have to, because it's 1. 1 is bigger than 0.25. So this is hypertonic. So if it's hypertonic outside, then water is going to do what? Is water going to move in or out? That's the question you have to answer. And you'll have to figure that out yourself, right? And so that's based on our lessons, and we can have a discussion about this when we when we meet again on Thursday or Friday, depending on which class is viewing this. So again, here's what we discussed in class. Summarize for you. We're looking at the net direction. We've used that word a number of times. Net is what's left over after you've subtracted and added everything. And you want to know what the net movement of the molecule is, of the water is. So you want to go ahead and draw a circle, or you can say, if you're typing it in the answer in the Nearpod, you can just write it's going out or it's coming in, if that makes it easier for you. So predict the direction of the movement of the molecule across the membrane by drawing two. So remember, what you got to remember is, in this case, you have to remember, you have to ask yourself, is the solute moving or not, right? And if you remember from class, you should know that the sugar is not going to pass through the membrane very easily. And so, and the reason is that it's large and it's polar, and so it's not going to want to pass through that membrane very easily. It needs a helper molecule that we'll talk about in our next session. So wherever the sugar's high, and this is just, there's two examples, right? And you have to figure out where the sugar is high, where the sugar is low. You're going to have to figure that out, right? So high and low is relative to which side. So it is relative inside to outside. Relatively high outside than inside, or is it relatively high inside versus outside? So it's either one side or the other. Inside the cell or outside the cell. Where is the concentration of sugar higher? So where is the number of sugar molecules compared to the number of water molecules higher? So where's the fraction, where's the number of the fraction sugar divided by water? Which one is higher? Sugar divided by water. The sugar is not going to pass through. So we know that because the sugar is polar and large, and that's not something that passes through the membrane easily. So because it's large and polar, it's not going to pass through. So as you know, if the concentration is going to be equal at some point, if you're going to try to reach equilibrium, if the molecules are going to be the same concentration on both sides no matter what, then you'd have to expect that the sugar concentration that where it's high is going to have water leaving out. So remembering that when you have high sugar, it's called hypertonic. I'm going to put the other word out here, hypertonic. So when we're talking about hypertonic solutions, if you remember correctly, water is going to move out, right? So water is going to move out of. Now, why is water moving out? Because the solute can't move. If the solute could move, it would move in. The solute would move in because it's higher. The solute's higher outside than inside. So the solute, the sugar, would move in if it could, but it can't. So the only way to get these two concentrations equal is to have water come out, and that's what happens. Water moves out, and this cell would shrivel, right? The cell would shrivel up. Okay? So there you have it. That's in general. That's what we're talking about here. Is you're talking about getting water. The movement of that water is called osmosis, and the diffusion of water is called osmosis. And it's moving from waters where water's high. And when you put these numbers here, you put sugar here and you put water here. You put sugar here and you put water here. Ask yourself, where's sugar higher? If it's hypertonic, the sugar is higher outside. If the sugar is high outside and low inside, and the water is the same, if the water is the same, then your concentration of water, the water is higher inside. So wherever the sugar is the highest, in this case outside, in this example, that's not to do with this example that's in your paper. You have to figure that out yourself. But in this example that I'm putting out for you here, the hypertonic's outside, so the water's going to move out. If the outside were hypotonic, in other words, the sugar was lower outside than it was inside, then water would move in. because water would be higher relative to the fraction that's inside. So when you do those percentages, when you do those decimals, and you find that the decimal outside is larger, the decimal number outside is larger, is hypertonic. The decimal inside is larger, then the outside is hypotonic. In other words, outside is lower than inside. And if they're the same, they're isotonic. And there's going to be there's no net movement. There's movement, but it's in both directions equally. Here, the net movement's out to the outside. And of course, if this were higher than this, the conditions would be hypotonic, and the water would move into the cell. And that would be if it were hypotonic, the water would move in the opposite direction. But this is hypertonic. Hyper, larger, greater, more. Tonic, solute. So there's more solute outside, where inside there's less solute. So if it was less outside, it would be hypotonic and water would go in. If they're equal on both sides, it would be iso. All right. Now someone did ask me a question on, and they said that this picture confused them. So this word crenated just means what happens when an animal cell is in a hypertonic solution. It's the same as plasmolysed. Both plasmolysed is happening in a plant cell, crenated is happening in an animal cell. That's when it's in a hypertonic solution, water's going to move out and the cell's going to shrink up, shrivel up. It's going to happen in a plant cell too. The difference is that when the plant cell has a cell wall, and the cell wall doesn't change its shape, just the membrane, the cell deflates, that's inside the cell wall, deflates. Then in isotonic solution, the cell retains its natural shape. So in red blood cells, it would be kind of an oval, like an inner tube. It has a kind of a dent to it. So if you're looking at a regular red blood cell with some from the side, it would look something like this, like an inner tube. And they're rounded if they're in their natural state, and they're tiny, but they're relatively big compared to bacteria, but small. They fit through your capillaries, and there's probably thousands of them millions of them at any given time in your blood stream. So all these cells, they're just floating around in your bloodstream, carrying oxygen to where you need the oxygen. All right, so that's their normal shape. They're oval shaped or the inner tube shaped if you looked at them from the side. When you take those same red blood cells and put them in a hypotonic solution, now water's going to rush in. They're going to turn very round and actually start to burst. If you put them in pure water, they'll literally start to burst. I wish we were in the lab. We would go ahead and take a look at what would happen by taking some cells and putting them into some distilled water and watching them burst open. Now, in plant cells, what would happen is they would become turgid. Again, they would become very rigid because water would rush in. Notice the arrows: water rushing in, water rushing in. Some water moves out because there's always water moving in both directions. Remember, this is all about random movement, this kinetic theory of motion, this kinetic theory of matter, that matter's always moving. So this water is moving in more than the water moving out, so this becomes rigid, stiff, straight, and we call it turgid. There's pressure pushing up against the cell wall. Here in this one, they're equal. They're equal movement in both directions. There's no change. There's no net change. Water's constantly moving in and out. Sugar's staying outside, but the concentration's the same on inside and out, so it's iso. In a hypertonic solution, there's more solute outside than inside, so the water is going to move out. Cell's going to shrink up. Notice the water moves mostly out. Some moves in, but more is moving out than is moving in, so the cell shrinks up. That's what this picture means. And so you go ahead and interpret the answers to these questions as you like. Now, I'm going to give you just a little bit of a clue here on this problem, number 25. Number 24, I think you should be able to do on your own. That's my feeling on this. But here in this paramecium is a single-celled organism. It's a eukaryote, has a nucleus. In these paramecium, they're what are called protists because they're eukaryotes, but they're also single-celled organisms. For the most part, protists are single-celled organisms. They have these. They need a contractile vacuole to help them. And when they use the word osmoregulate, right, keep osmotic balance, they mean you need to be able to keep. You have a cell that doesn't have a cell wall. So if it swims along and finds a condition that is hypotonic outside, how is or hypertonic? How could this eukaryote that has a bunch of different rooms use a contractile vacuole? The clue is in the word contractile. In other words, it can squeeze or expand. How can it use that contractile vacuole to help it keep the concentration of solute in here the same? We'll talk about it when we meet in class, but this is actually very much a challenging question, and you have to think about what a contractile vacuole would do. All right.